Phosphorylation and dephosphorylation do not play a role in ligand-gated ion channels. Phosphates are not involved in ligand-gated ion channels, so the addition and removal of them is not applicable to this process.
When calcium ions enter a cell via ligand-gated ion channels, its presence triggers a certain reaction that causes the neurotransmitter in that cell to leave to the polysynaptic neuron via exocytosis. There is a high concentration of calcium ions outside of the cell, so the calcium ions enter the cell because of the need to travel from high to low concentrations through passive transport, down the concentration gradient. Once they are present in the cell, they trigger the neurotransmitter in that cell and its synaptic vesicle. Serotonin is the neurotransmitter. The neurotransmitter will undergo exocytosis. The synaptic vesicle opens up and becomes one with the cellular membrane, releasing the neurotransmitter serotonin out of the cell. Serotonin will then bind with the neurotransmitter receptors of the polysynaptic cell, and that will initiate a signal to the cell body of the polysynaptic neuron. Overall, the calcium ion's presence is what signals the movement. These ions are called secondary messengers because they are transported into the cell as a result of the ligand binding to the allosteric site, which takes the role of the first messenger. So, the word secondary indicates that their function is only triggered after that of the primary messenger. They are classified as a messenger because their presence in the cell sends a message to do something else. In this case, it messages the serotonin to leave the cell via exocytosis, ligand-gated ion channel.
At rest, calcium ions are maintained at high concentrations outside of the cell, so cells can easily access them to pump them into the cell. When activated, ligand-gated ion channels allow neurons in the brain to rapidly increase their calcium ion concentration in the cell's cytoplasm by opening up channels, the ligand-gated ion channels, within the cellular membrane. Ligand-gated ion channels allow the cell to maintain homeostasis because the cell signaling is regulated, and so will open up these ion channels when needed. And a need for serotonin, for example, will allow the channels to open up, and serotonin will leave the cell as a result of the calcium ion's presence. Homeostasis is the state when cells are balanced and equal, so the ligand-gated ion channels allow for this to happen to maintain balance. When activated, ligand-gated ion channels allow for calcium ions to be pumped into neurons of brain cells and transmit secondary signals. Also, on a broader scope with cell signaling, cells can communicate whether or not they are at homeostasis. If the body is not at homeostasis, cells can communicate where in the body the problem needs to be fixed with the cell signaling to regain homeostasis. This is effective because through cell signaling, the problem can be pinpointed in the body and cells can communicate what specifically needs to be done in that part of the body.
Homeostasis is keeping cells balanced so that they can function correctly. The phospholipid bilayer of the cellular membrane is semi-permeable, so the membrane can allow certain materials to pass through, aiding in homeostasis. Cells can also undergo mitosis if more cells are needed for a certain process, which aids in the maintenance of homeostasis. And finally, plant cells have chloroplasts, and both plant and animal cells have vacuoles that work to maintain homeostasis in cells. Chloroplasts use the sunlight's energy to convert into sugars, fueling plant health and growth. Without chloroplasts, plant cells wouldn't have a stable income of energy and would die off. Vacuoles regulate cell pH through the amount of hydrogen ions. They also maintain amount of turgidity based off of pressure. How do vacuoles help maintain homeostasis? All of these factors contribute to how cells maintain homeostasis. How does your cell membrane help your cells maintain homeostasis?